Hello everybody. Today we're going to talk about Gregor Mendel. He's the father of genetics, or considered the father of genetics. He was born in 1822 in Austria. Um, Later in life he became a monk, and during his life as a monk he was very interested in heredity, the way how things were passed on from one generation to the next. And when he was studying heredity, he used the pea plants because it was a very quick turnaround. He gets seeds very fast. He could plant them, get data. So it was very nice to work with these pea plants. And let's take a look at why. So if we take a look at the characteristics of the pea plant, we're not really looking at too much. We have the seed shape, seed color, pod shape, pod color, flower position, and stem height. Not too bad to work with. But what makes it super nice is that for each of these characteristics, there's two possibilities. If we look at seed shape, we only have either round or wrinkled, or for seed color, yellow and green, or purple and white for flower color, and so on. So it made it very easy to see how things were passed on from one generation to the next. And when he did this, after doing months of or years of observations, he came up with a few laws. The first one, which we're going to take a look at now in this video, is the law of dominance. He noticed that as certain traits moved on from parents to their offspring, that some traits would be visible right away, some would be lost. So he came up with this law of dominance. We know today that this law of dominance is referring to genes or DNA, but he didn't know about DNA or genes at that time, so he called them factors. Basically, he said there is some factor or some item that is getting transferred from the parents to the offspring, and depending which one of those is dominant is going to be the one that shows in the offspring. So let's see how he came up with this idea of law of dominance. Get to the next slide. All right, so what he did was he took a tall plant and a short plant. Since he started off with these plants, he called these the parent. All right, and these are the parents, got their seeds, planted the seeds, and waited for them to grow. And he called this, since these are the offspring to the parent generation, he calls these the F1, or first filial. And he noticed that 100% of them were tall, no short. Well, you must think, well, here we go. Here we had one parent that was tall and one was short. You mean to tell me that 100% of them were tall? What happened to this short characteristic? Was it lost forever? Would it come back? So there's only one way to figure that out. And what he did was he cross-pollinated the offspring, or the F1 generation. He cross-pollinated them, got their seeds, planted them. Now the offspring to the F1 is known as the F2, or the second filial. And he noticed that the offspring, or I shouldn't say offspring, but the seeds developed tall and short plants in a 3-to-1 ratio. For every 3 tall plants, there was 1 short. So that short characteristic came back. So that's how he came up with the law of dominance. After thinking about this, he said, all right, well, here we have a tall plant, here we have a short plant. That tall and short factor, remember he didn't know about genes, combined in this group, in the F1. But since they're all tall, that means that the tall gene, or that tall factor, must be dominant. And then when he crossed these, that short trait must have come out to produce a quarter of them as being short. So he developed a Punnett square to show this. So let's see. Go to a blank page. Clear the board. All right, so what he did was he made a grid. Now he had a short plant. Now he said that short is recessive, tall is dominant. So first thing, we represent the tall characteristic with a capital letter. So we'll say, and we usually use the first letter of the dominant trait. So since tall is dominant, we're going to use capital T. So T is tall. And then the lowercase t is short. So based on his studies, he said, all right, so in order for a recessive trait to show, it must have two recessive traits. So we'll do that. The tall trait, we have the tall plant, he said, was big T, big T. And then when he did the first cross, now what you see in this box, what I'm doing, are the gene makeup of the first filial, or the F1 generation. So if you notice, when we do this cross, we have 100% that's tall, right? Because when you have a big T with a little t, or a dominant trait with a short trait, the dominant trait shows. And then he took this first filial, what's in these boxes, and he crossed them. Now if you notice, all of them are big T, little t. So let's make another box here. So you have big T, little t, big T, little t. And when you cross these, you'll notice that you have 75% is tall and 25% is short, or a 3-to-1 ratio. So my question to you is, it's pretty obvious that when you have big T, when you have a short plant, it has to have this makeup, little t, little t. So my question to you is, how would he know a tall plant? How would he know if it was big T, or big T, little t? What kind of experiment or test would he have to do on the plants to figure out the genetic makeup? Now, before you answer that question, what you might need to do is look up these terms. I want you to look up genotype, phenotype, homozygous, and heterozygous. So I want you to look up those 4 terms, and I want you to figure out, or find out, how Gregor Mendel was able to figure out for that parent generation, that P generation back here. Let's go back to that slide. How did he know that this plant here was big T, not big T, little t? Okay, what did he have to do to figure out what this is? Because you can't tell by looking at it which one it is. All right, so I hope this was helpful, and we'll talk about this tomorrow in class. Have a good night, everybody.